My name is Namita Sahai from Crystal Springs Upland School, and I will be presenting my project that works to treat disorders including heart failure, hypertension, atherosclerosis, cardiohypertrophy, and stroke that affect millions of people. Now, what do many of these disorders have in common? They are caused by clotting and obstructions. By controlling vasodilation further, we can find treatments for not only the highly rampant disorders, but also some of these rare ones like Moyamoya disease, which has little to no treatment options. The first goal is to find a target to later develop drug candidates for. My project targets soluble guanylyl cyclase because it is a site that converts GTP or guanosine triphosphate to the signaling molecule cGMP, which controls vasodilation. An integral part of this project is to uncover novel structural information about this protein using evolutionary biology techniques. To start off, in order to get the structure for this protein for humans, I have used AlphaFold 2 software. What AlphaFold does is uses artificial intelligence, as it is a deep learning system, to take in amino acid sequences and perform predictions on protein structure. The network predicts interactions based on structural and genetic information, which culminates to a final display after working part by part. Confidence scores are calculated to display the most accurate parts of the protein. After gaining this structure, I needed to figure out what the conserved residues of the protein are most integral for its function, thus finding the most druggable parts of the protein. Doing this involved looking at different species to determine which parts of the amino acid sequence stayed constant. Any similarities would most likely be attributed to functional or structural importance. It also suggests low mutations across multiple species as humans have evolved. To find the conserved residues, I use multi-sequence alignment analysis, which involves collecting homologs, inferring MSA, and reconstructing the phylogenetic trees showing evolutionary relationships. An MSA is just a homology of multiple sequences of the same length. A similar homology modeling is done on hemoglobin, as soluble guanylyl cyclase has a heme binding site that is also present on the protein. I hypothesized that SGC would not be a good target if the two structures were too similar, as any drugs would negatively affect the oxygen and iron in blood as well. Fortunately, there was only 11.92% some identity similarity. They had vastly different interactions under a superimposing analysis. With the evolutionary information in mind, the HNOX domain was proposed as a target site, as it includes the heme binding site as well as a majority of the conserved residues. A library of thousands of ligands were collected from Zinc-15 to find drug candidates. In the end, a couple hundred had the best properties and were run through molecular dynamics. Each ligand went through density function theory optimization to avoid the effects of expected forces and the delta G values, and binding affinities between ligands and receptors were collected. After finding out which of the ligands had a high affinity for SGC, I analyzed ADME toxicity properties such as Lipinski's Rule of 5 and Brank's filters. They make sure that the ligand is suitable to be a drug, has good absorption, and low toxicity. The ligands with the best performance included C15H24N4O, C9H11NO, and C16H20N4O4. Finally, the similarities between the top performing ligands were noted, such as alcohol groups and amine groups. Some future directions would be expanding the variety of ligands based on drug parameters previously outlined, as well as validating the results through mice trials. Ultimately, determining the areas of protein sequences that may have shown mutation bias across a phylogenetic tree may help to develop drugs for other targets as well. Finding effective drug candidates can not just treat cardiovascular disorders causing up to 31% of deaths worldwide, but also likely have a role in cancer regulation, major depressive disorder, and more. Thank you so much for listening to me.